Hello students, we're going to have a little lesson in genetics today to help you with your lab. I'm going to talk about some terms first, and then I'm going to show you how to map out Punnett squares. So DNA. DNA is the molecule of heredity. It contains all the instructions that are needed for any organism to have the traits that that organism needs in order to survive. Now, the DNA is comprised of different segments, and these are called genes, though not all of the DNA contains a gene. The genes are the separate traits, the code for the separate traits found in the DNA. So you can have a gene, for example, for eye color. There are different versions of this gene, though, and these are called alleles. So you can have eye color genes that are blue eye color genes, or green eye color genes, or brown eye color genes, etc., etc., etc. Hazel, etc. So alleles are the different versions of genes. If you're an organism that does sexual reproduction, which of course humans are, then you'll always have two genes for every trait. So you have two genes for eye color. And how do we know this? Because you get one set of genes from your mother and one set of genes from your father. So you always have two copies. Now, you might have two different alleles present. So your mother might have contributed a blue eye color gene, and maybe your father gave you a brown eye color gene. And there are lots of different combinations, of course. So the types of genes that you have present and the types of alleles that you have present are called your genotype. So this means what two genes are present in you. All right, this is a nice question to ask, but this is a little bit hard to see because you can't see which genes you have. You have to do molecular analyses in order to figure this out. But you can see phenotypes. So phenotypes are the physical traits that you can see. All right, so these are easier to determine because you can actually see these. Now, everybody knows, I know that you know this already, that there are dominant traits and there are recessive traits. A dominant trait you will always see expressed in the phenotype, always. Whether you have one copy present or if you have two copies from both of your parents, you will always see a dominant trait being expressed. Recessive traits, you only see sometimes. Sometimes expressed as a physical trait. And the only time that you can see recessive traits is if there are two recessive traits present, because a dominant trait will mask a recessive trait. Okay, let's move on to how to do a Punnett square and how we write these different traits. And we're going to just use A's to start with. So we're going to have a capital A and a little a. And so A, whether it's capital or lowercase, tells you that this is a particular gene. So say it's the gene for eye color. Okay, so we're representing eye color by A's. Capital A tells you that this is a dominant allele. You use a capital letter to show you that it's bigger and more powerful. And so in our example, capital A could mean brown eye color because that is dominant over blue eye color. Little a could represent a recessive type of eye color, and that would be blue eye color. Okay? So now you have two parents, right? Say they both have brown eyes. Let's see how it's going to be, how their children are going to turn out. there are a couple of ways that we can look at this. If you have two brown-eyed parents, then they can either have the genotype capital A, where they have two brown eye-color genes, or they can be capital A, little a, where they have a brown eye-color gene and a blue eye-color gene. Okay? Now say that one of your siblings ends up with blue eyes, and everybody in the family is shocked. Is there any way that that could happen? Let's see if it's possible. Let's assume that they are both of your parents are capital A. So capital A mated with capital A. Okay. This parent right here can only contribute a capital A allele to any offspring. And the same for this parent over here, capital A. And so if you put these two together, all of their children are going to have the genotype capital A, and that is all brown eyes. Oops. So if that's their genotypes, then they can't have children with blue eyes. So what if one is capital A, and the other one is capital A, little a? And this is called heterozygous. And that just means you have two different versions of the gene. Okay? All right, what can this parent contribute to any offspring? Just a capital A trait. What about this one over here? Oh, capital A or a little a. All right, so here's where we can make a Punnett square. So we've got a capital A on this side representing that parent. And we're going to make a big block like this. And put that A right here and the little a right there. And so let's see what kind of offspring we make. Capital A, little a. Okay, what do all of these offspring look like when you see them? What are their phenotypes? Brown. Oops. All brown-eyed children. Uh-oh. All right, let's keep going. Keep using my example. Look at all my little black dots. Now, what if your two parents are heterozygous? Right? Meaning they have both kinds of traits. Brown eyes and blue eye colored genes. So this parent can contribute either a capital A or a little a to an offspring. This one, same thing, capital A or little a. All right, so let's set that up in a Punnett square. We put all the possibilities of genes from one parent down one side and all the possibilities from the other parent on the other side. The reason I'm drawing circles around these are because these actually represent gametes or eggs or sperm. So they're the sex cells, because sex cells only have a single copy of genes in them. Okay, so now let's put together all these different possible combinations. Capital A, little a, capital A, little a. You always put the capital letter first, and then little a, little a. Aha! Okay, let's see what we have then. What's this? Brown. Brown. 1, 2, 3. 3 browns to what? Woo! Blue. 1 blue-eyed child. Okay, so the ratio, oops, there you go. The ratio of brown-eyed children to blue-eyed children would be 3 to 1. So there's 3 chances out of every 4 that your children would have brown eyes, but you can still have a child with blue eyes. So that's how you can have a child with blue eyes produced by parents who have brown eyes. Eye color is actually a little more complicated than this, but I'm just using this as an example to show you how this works. Okay, in the crosses that have been done for us by Carolina Biological, we have some plants that have been crossed. We have the let me get a clean sheet of paper. We have a parental plant, P1, and that's what we have. We also have a mystery P2 plant, so these are the parents that you're starting out with. Okay? We have parent 1 and parent 2. And this is what is called a dihybrid cross eventually. Di means 2. So we're looking at 2 different traits. So I'm going to write those over here. We have purple stems on our plants and yellow, green leaves. Okay? Just going to scoot that over just a little bit. So for purple stems, we're going to represent this by P's. A capital P and a lowercase p. And so you're either going to have purple stems or white stems. But we're going to represent that with P's because that's the color of the stems. Then we have yellow-green leaves. Let's represent this with Y's. So capital Y and lowercase y. Make sure that you make a distinction between your letters so that I can tell what they look like. Okay, capital Y, little y. So you can either have yellow-green leaves, which are light-colored, or you can have dark-colored leaves, which are either dark green or purple. Okay? Now, what we want to know is, what are our 2 parents that we start out with here? And we've got to have P's and Y's in their genotypes. Okay? Now, we have been given the information that these are true breeding homozygous parents. True breeding homozygous parents. True breeding means that you can cross individuals in this P1 generation, any of the P1 plants, and you get the same offspring time after time after time after time. There is no variation. So this is what that means. You're either going to have 2 capital P's or 2 little p's in the genotype. You've got to have either 2 capitals or 2 little p's. And the same thing goes for the y's. Okay? You can either have capital Y, or little y, little y. Now the thing is, you don't know whether your parent is capital P, capital Y, or capital P, little y, or little p, capital Y, or little p, little y, little y. And you don't know whether purple stem is the dominant trait or white stem is the dominant trait. You don't know whether yellow leaves are a dominant trait or yellow leaves are a recessive trait. So this is what you got to figure out with the Punnett squares that you're going to be producing. And you also don't know what your parental 2 is, but they also have to be either this, or this genotype. Okay? So the first thing that you have to do is figure out your P1 generation. So look at them and see what types of traits show up.